Hello everyone, welcome to Nature Narration webinar series by Aghanaashini Nature Foundation. Today's webinar on citizen contribution to Shieldtail conservation by Dr. Vivek Philip Cyriac. Dr. Vivek Philip Cyriac is an evolutionary ecologist with a broad interest in understanding processes generating and maintaining biodiversity. His work centers on understanding how environmental factors and biotic interactions influence speciation and create biogeographic patterns. As a herpetologist, he has spent over a decade studying shieldtail snakes, one of the most poorly studied snakes in India. He is also one of the founder of the Shieldtail Mapping Project, a citizen science initiative focusing on the study and conservation of shieldtail snakes in India. Now, the session is handed to Dr. Vivek Syriac. Dr. Vivek, I request you to take over the session.
Okay, thank you. Yeah, I hope my slides are visible here. Right. Okay. Um, cool. Thank you for that introduction. Yeah, hello everyone. Thank you for joining me today on this webinar. So today I'm going to talk to you about tree-tailed snakes, which are a group of snakes that has fascinated me for a very long time. So I'm going to try and sort of summarize the work that we've done with tree-tailed snakes over and also talk to you about the role of citizens or the general public and how they've been instrumental in understanding a lot more about shieldtail biology and also towards making conservation assessments. So, as I said, I'm a herpetologist, so I mostly study amphibians and reptiles. And one of the reasons why I'm so fascinated by reptiles is because they're exceptionally diverse, right? They are the most diverse group of land vertebrates. So if you look at the total number of reptile species, there are more than 12,000 plus reptile species. And this number is constantly increasing every day. Right. Every single day, there are new species of reptiles being, discovered every day from all across the globe. Right. So let me just try to get it. Okay. And this is something that has fascinated. And it's not just species diversity. There's a lot of conservation challenges as well when it comes to reptiles. Right? It's said that 1 in 5 species of reptiles are threatened with extinction. Right? And so if you sort of look at these plots here, they just show the number of endangered and critically endangered and vulnerable species and so on. But you'll also notice that when it comes to reptiles, there are a lot of species that are data deficient. That means there is no data available to actually assess the extinction risk of these species. Right? And if you look here, you'll see that this gray sort of region is the data deficient species, the number of data deficient species. So if you look towards on this plot over here, you'll see that most of the data deficient species come from this group called as squamates. Right? And squamates are basically just lizards and snakes. Right? And lizards and snakes are the most diverse group of reptiles. In terms of their morphology, they come in all sorts of shapes and sizes, from small little, blind snakes and geckos to huge anacondas and Komodo dragons, right? But they also occupy several different kinds of habitats. There are many species that are arboreal, that means they're adapted to living on trees and in the canopy. And they also have their own set of adaptations. For example, you can see this Draco over here with these, patagium that they use to glide. There are quite a few species that are aquatic and have their own set of adaptations. A large majority of the species are terrestrial, in the sense they move around on the surface of the earth. But quite a few of them are fossorial, right? Fossorial meaning these species dig into the soil, burrow into the soil, and spend most of their time living underground. And this is the group that has fascinated me the most because I know so very little about these species. Right? Now, fossorial species also have their own set of adaptations. Right? So burrowing into the soil is one of the most energy expensive behaviors. So if you look at this plot here, this line represents the cost or the energy required for walking or running. And you see that burrowing, that is all these points, the energy required is much higher than just walking or running. Right? And because of this, fossorial species occupy a very unique morphospace in the sense they look very different from all other ecological guilds. Right? So if you look at a burrowing reptile, what you notice is that they have a very small, hard head, a super elongated body, and a small, short tail. Right? And this is what we call as the burrowing ecomorph. Right? And this is opposed to what you would see on the surface. Right? So a surface-dwelling snake reptile would have, say, a much broader head, a less elongated trunk, and a really long tail, which they sort of use to sort of navigate on the surface or on trees and so on. Right. And interestingly, we also have fossil evidence to show that every time a lineage becomes fossorial, they end up taking this sort of form. Right? For example, this is a group of lizards that we call as amphibians or mole lizards. These are all fossorial lizards, and they're burrowing lizards. And so you see most of them don't have any limbs. But there's this very interesting little species which sort of has two little limbs sticking out just behind the head. Right? And recently, researchers have found several fossils within this group, okay, which show that they had legs, right? And based on these fossils, they've sort of reconstructed that every time a reptile ends up becoming fossorial, what happens is shortening of the limbs, an elongation of the body, shortening of the forelimbs, and then finally you end up looking like this. Right. Now, while this morphology allows these fossorial species to efficiently burrow into the soil, they also make them super specialized to just living in the soil. So if you take a fossorial lizard and leave it on the surface, they're quite clumsy, right? So these fossorial species, the fossorial reptiles, tend to be much more specialized than other reptiles. Right. And such specialization can also be a problem. So extreme specialization can restrict the ability of species to sort of adapt to environmental fluctuations, right? And this in turn can increase your chances of extinction. So if you look at this sort of graph over here, you'll see that species that are more specialized, that are closer here, tend to be more vulnerable to extinction. Being shown in many different groups, such as birds, bats, trees, insects, and so on. Right? But how do you assess extinction risk? Right? One way to do this is to look at how species have responded to environmental changes in the recent past. Say, you can look at how species are responding to climate change in the last 10 years or 20 years and so on. Right? And then you can see whether the species are adapting to this change, or you might see that some species are changing their distributional range, sort of following this environmental conditions wherever they go. Or in extreme conditions, you might actually see population shrinkage. And this is very concerning, right? So when you see population shrinkage, it means that there is a chance that these populations can go extinct. Right? But doing something like this to sort of get is really difficult, right? We might be able to look at larger mammals which are more charismatic and more easily visible and so on. But when it comes to reptiles, which are very cryptic, very hard to find, getting the data required to actually look at how species are adapting to environmental change becomes really difficult. Right? So what do we do? Another way of assessing extinction risk is to actually go back into the past, right? Look at how species have responded to environmental changes in the past over millions of years. And based on this, you can sort of make inferences on how species might respond to, say, present-day climate change or environmental conditions in the future, right? So how do we do this? Of course, there are many ways of doing this, but one way is to actually reconstruct the evolutionary history from DNA, right? So what we do is we take right? And based on your DNA, we build what is called as a phylogenetic tree, right? So this is basically just an evolutionary tree which shows relationships between different species. And now we have methods to date this tree, right? So now we can say when, say, a particular lineage evolved, how many millions of years back that it evolved, and so on. Right? Now, whatever tree we build, right, this particular tree that we see here is the result of two processes. One is speciation, which is the formation of a new species from an ancestral population. And of course, we have extinction. Extinction is basically removal of a species or a branch from the tree, right? And this is what we call as diversification, right? And this is what has led to literally every species that we see on the planet today, right? So what we can do is we can actually reverse engineer these processes, right? Just by tree like this, right? So we can reverse engineer what the speciation rate would have been, what the extinction rates would have been, and so on. Right? And so that's sort of what we did, right? So we asked the question whether the burrowing snakes are at higher risk of extinction, and we sort of published this paper in 2018, where we calculated these diversification rates. Right? And what we see is that fossorial species tend to have a lower speciation rate, but a much higher extinction rate. Right? So what this means is that fossorial snakes, just because of the specializations that they have, they just tend to be more vulnerable to extinctions than other groups of snakes, right? But even though fossoriality has these evolutionary consequences, we see that it's still widespread in snakes. So all these black lines are fossorial species. And if you look at this tree of snakes here, you see that there is one large group of fossorial snakes, which we call as the blind snakes, the worm snakes, the thread snakes. We have quite a few of them in India, right? And we have one more group, large group over here, which we call as the Asian annelids, right? And this Asian annelids have two groups. One group is called as the Shieldtail snakes, and the other group is called as pipe snakes, which we find in Southeast Asia. And this is the group that I have been interested in, right? And I'll be talking to you more about Shieldtail snakes. Shieldtail snakes belong to this family called Europeltidae, right? They're small size snakes. They just grow yay big, which is all usually less than 2 feet in length. They're all non-venomous. Presently, there are 7 genera, right, and a total of 73 species, right? These are all ovoviviparous snakes in the sense that they give birth to live young, and they're all fossorial. They spend most of their time living. And if you look at Shieldtail snakes, they have a very peculiar body morphology. The face is more pointed, and the tail is very short, and they have this blunt, stubby tail. And many species have these hard keratinized scales, which sort of form a disc-like structure, and that's why they get the name Shieldtail snakes. Now, these snakes are distributed only in peninsular India and Sri Lanka, and they're largely restricted to the wet zone. So most of the diversity is in the Western Ghats that's around here. We have a few species in the Eastern Ghats here and a few species in Sri Lanka. And these are insanely diverse snakes, right? They come in all sorts of shapes and sizes and different forms, of course. But what's more fascinating is the colorations. And these are amazingly, just absolutely gorgeous looking snakes, right? So we have super iridescent snakes like this. There are many species that are golden yellow and yellow, some that have a purplish tinge, a few species that have bright red or scarlet colorations. They're absolutely beautiful looking, right? And what's even more fascinating is that they have all these colors, even though they live underground where there's no light, right? So the evolution of colors itself is an super interesting question when it comes to Shieldtail snakes. But of course, we won't be going into that. But apart from this diversity, they also have a lot of specializations. Right? Now, Shieldtail snakes, like all other burrowing reptiles, they use their head to burrow into the soil. Right? And because of this, they have extremely ossified skulls. So the skull is one compact unit. And this is very different from other snakes that have a lot of space in between their skull elements. Right? And so this ossified skull helps them burrow into the soil, and you'll find shield kinds of head structures. Some have a head that is more shovel-like in shape. Some are slightly more pointed, and some have this extreme pointiness. Right? Of course, we don't really know why they have such diversity in their head structures, but it is likely associated, with sort of burrowing in different kinds of soil. Right? Now, because they have these extremely ossified skulls, there's not a lot of space for jaw adductor muscles. And because of that, they're restricted to eating prey which are soft body. Right? So almost 95% of their diet is just earthworms. Right? So there's specialization in their diet as well. There's also specialization in the way they move. So how they burrow into the soil is basically they stick their heads into the soil, they contract the first one third of their body or the spine, and then they ram their head into the soil, right? And that's how they sort of burrow. This kind of specialized locomotion is called as a freight train locomotion because it's sort of like a train, right? Where the engine is in the front and the rest of the body is just sort of dragged along. Right? Now, because they have this very specialized locomotion, they also have specialized musculature. Right? So the first one third of the body is packed with red muscles. Red muscles have a lot of mitochondria, which produce energy for sustained work. Right? And the rest of the body is sort of flabby with white muscles. So if any of you all have ever held a shield tail, you'll notice that the first one third of the body is really hard and strong, while the rest of the body is all like flabby, right? So all of these specializations again make them very, very restricted to the sort of environment that they are adapted to. Right? So you take them out of their environment, the shield-tailed snakes are very, very clumsy and very uncomfortable. Right? So we wanted to see whether, what is the extinction risk for shield-tailed snakes, given that they also have so much of specialization. Right? So again, we went back, we built an evolutionary tree for these snakes, and again we calculated speciation rates, extinction rates, and so on. Right? And what we saw is that every time there was sudden fluctuations in your environment, rather temperature, we saw an associated increase in extinction risk. Right? So if you look at this red line here, this red line tracks the temperature, model temperature over the last 55 million years, right? And here we have extinction probability, and you see that around 50 million years, there's evidence for positive high extinctions, right? And if you look at this 50 million years is when the temperature was extremely high, right? Again, at around 30 million years, there's high extinctions. And when you go here, you see around 30 million years is when the temperature dropped down all of a sudden, right? So whenever you have these sudden fluctuations, you are seeing an associated increase in extinction rates. Right? So what we showed is that these snakes are highly sensitive to environmental changes. Right? So much so that even above ground temperature fluctuations can influence the evolution of these snakes. Right? So this just raises the conservation concerns, right? Especially given the rate at which climate is changing currently. Right? So, but if you look at the conservation status of shield-tailed snakes, right, most shield-tailed snakes, more than 17 shield-tailed snakes are not even assessed. So IUCN is what basically is the body that sort of assesses the conservation risk of different species. And they classify them into endangered, vulnerable, and so on. Right. If you look at shield-tailed snakes, almost 22 species of shield-tailed snakes are considered threatened. That means they're either in vulnerable or endangered or critically endangered. Right. But what's more concerning is that more than 40% of shield-tailed snakes are data deficient. That means there is no data to sort of assess what the extinction risk of this snake is. Right? And this is because there are several challenges to data collection itself. Fieldwork, shield fieldwork is extremely challenging. So most of these snakes are found in thick, dense forests. So we have to go, especially during the monsoons, we have to go to these forests and basically dig for these snakes. Right? And where do you dig? right? Because there's so much of land where you can dig, right? So it's sort of like a needle in a haystack situation. So one of the things that we do is we lift huge logs and rocks and we dig under these. But again, finding a shield tail is still very hard, right? Even after digging and lifting these huge logs and digging, many a times we come back without any samples, right? So fieldwork is super challenging, right? And so the amount of data that we can get is again very limited. Another challenge to shieldtail conservation is just the complicated taxonomy. Right? Most of the shieldtail species were described in the late 1800s during the colonial period, right? And this is when there was not a lot of methods like genetic methods, DNA methods, and so on and so forth. Right? And this has led to a lot of taxonomic complications. So there are many species that have been clubbed together and called one species, while on the other hand, you have the same species that have been split and called as different species, right? So here's just one example. It's called rhino.
Sir, how do you know where we have to dig for this, sir?
Oh, that's just luck, yeah. I mean, over the last 10 years, intuitively, we've gotten better at it, but, largely it's just luck. So what we do is we look for logs like this, which are sort of decaying. And so under these logs, you have slightly more humusy soil, right? So that's where we dig, but there is no guarantee that we will get these, we will see these things, right? So I was talking about the complicated taxonomy. So here's just one example of the species called Rhinophis sanguineus, right? And this was described by Colonel Richard Henry Beddoe somewhere in the late 1800s. And this is the specimen with which he describes the species. So this is the holotype of Rhinophis sanguineus. The same Richard Henry Beddoe described another species called Rhinophis microlepis, right? Until now, we didn't know for sure what this species was, but after a lot of work, now we know that the species Rhinophis microlepis is just a juvenile of the same Rhinophis sanguineus, right? So there is so much of complications, right? And because of these complications, many of the samples that we've got, we haven't been able to identify to the species level as well, right? It's been a huge challenge just to identify species. Right. But yeah, we're making quite a lot of progress on this end because with recent advances in DNA sequencing technology, we're now being sequencing DNA and so on. And we're also collaborated with the Natural History Museum, and they were recently able to sort of extract DNA from 200-year-old specimens in the Natural History Museum in London, right? And now that we've combined both of these data sets, we've been able to build this large, nearly sampled phylogenetic tree of shieldtail snakes, right? And what this exercise has led to, apart from clarifying a lot of these taxonomic issues, is we also understood that shieldtail diversity is hugely underestimated, right? So when we started this work, there were about 56 odd species. Currently, there are 73 known species, but we estimate that there could be at least 85 to 100 species of shieldtail snakes. And this makes shieldtail snakes one of the most diverse groups of snakes in India. Right. Now, just to give you a perspective of how important this is, shieldtail snakes belong to this large group of snakes called Haenophilia, which also includes boas and pythons, right? Now, boas have a global distribution, boas and pythons, right? So pythons, pythons have a global distribution. They're found, say, Africa to Australia, but then there are just 38 species. Boas also are found all the way from Southeast Asia to America, right? But there are just 67 species. If you look at shieldtail snakes, they're restricted to just peninsular India and Sri Lanka, and there could be more than 100 species within shieldtail snakes. Right? So there's something really interesting happening with shieldtail snakes, especially from an evolutionary perspective. Right? And we're also finding a lot of cryptic diversity, right? Which basically means that there are snakes that look exactly the same. Morphologically, they're not indistinguishable, but genetically, they are very distinct species. So this is an example of a group called Territorus, right? Initially, it was thought to be just one species, and then later on, researchers came and said that there are four species. Uh, Recently, just last year, we published this work where sampled across the Western Ghats, and we showed that there could be at least 10 species within this group, right? So there are many species which are morphologically very similar but are clearly different species, right? And this has, of course, led to us describing several new species to science. This is just two examples of rhinophis species from Wayanad, which we discovered in 2020. Just last year, we described four new species of territorials from across Kerala and um, and Karnataka, right? And there's still a lot more species that are yet to be sort of described from the Western Ghats and Eastern Ghats. Right? Now we've sort of reached the stage where we're able to identify many of the species. We're getting some more taxonomic clarity. But what about geographic data? Right now, unfortunately, we know what these species are. We don't know what their geographic range is. Right, and this data is very, very important because when it comes to reptiles, one of the main criteria that IUCN, that is the International Union for Conservation of Nature, right, and this is the body that classifies species as endangered and so on. Right, one of the main criteria that they use is geographic range. Right? So if you don't have this information, right, we don't know what the conservation status of these species are. Right. So if you look at the global distribution of reptiles, right, these range maps that I was talking about, there are two bodies that actually maintain such distribution maps. One is, of course, the IUCN Red List, and then there's another body called the Global Assessment of Reptile Distributions, which maintains these range maps. So how are these range maps made? These are made by experts, right? So what they do is of these different groups, and these experts come in, and based on their opinion, they draw a map of where each species is sort of found, right? And this expert-drawn map has been used widely by different researchers to address different conservation questions, and also published some, really high-impact papers in high-impact journals, right? But the problem is that to a large extent, we take these expert drawn range maps to face value, and we don't question the accuracy of these maps because, of course, it's experts like us who have gone and drawn these range maps, right? So here's just an example of what an expert drawn range map looks like, right? So this is an example of king snakes, and if you look at this one species of king snake, say desert king snake here, and these green dots are where all the points where they've actually seen the snake, right? But then, these are the two experts, Alex and they worked on the snake for a really long time, and based on their expert knowledge of their natural history, they say that this snake should be found all over here, even though there are no actual points here, right? Well, this kind of range map is still very, very important because if you don't include these ranges, you're going to fail in your conservation assessments, right? But then when you build these range maps, you still need to have an in-depth understanding of that species, its ecology and its natural history and so on. Right? So we wanted to see, okay, how well are these Shieldtail range maps, right? So what we did is we downloaded all the range maps from IUCN and GUARD, and we sort of overlaid it onto each other. And what we saw is that these range maps don't really represent the range of Shieldtails. So you can see these are places in the Western Ghats where it shows that there is no Shieldtails at all, right? But we surely know that there are Shieldtails here. I mean, this is Wayanad, which is one of the hotspots for Shieldtails, right? Same here in GUARD, you'll see that again there are gaps like this, but you also see one really small red dot here in the center, right? And this red dot is somewhere in between near Munnar and Valparai, right? And this is literally a herper's paradise, right? Every herpetologist wants to go there and and do herping and so on. And so you end up having huge biases in the sample, right? So there's clearly problems with these maps. And the reason is because even though we are experts at drawing this map, the amount of information that's available to the expert itself is very limited. Right. So there's clearly a need for data-driven sort of range maps. Right. And because of this, to get such data, we started the Shieldtail Mapping Project. So this project is sort of intended to be a citizen science initiative, which 2021. And as part of this project, we've been doing a lot of outreach and public engagement and so on. And in turn, we're getting information from the citizens and from public, right, who've been going hiking or doing whatever, right? And we're getting that data and we're using this information to make better conservation assessments, right? So there are three main goals of the Shieldtail Mapping Project. One of the goals is to train local naturalists, or what we call as Shieldtail ambassadors, and I'll talk about this a little bit. Another goal is to bring awareness to different communities, mostly focusing on different plantations and estates in the Western Ghats and Eastern Ghats, and then ultimately using all of this data towards Shieldtail conservation. Okay. So, like I said, one of the main things that we have is what we call as the Shieldtail Ambassador Program. So, Shieldtail ambassadors are they can be naturalists, can be local leaders, students, Shieldtail enthusiasts, anyone who's who's fascinated by Shieldtails. Right? And what we do is we train these ambassadors, and what we expect from these ambassadors is that they go about documenting Shieldtails in their local areas, and also help us plan and conduct awareness programs within their local communities. Right. So, in a way, ambassadors are basically liaisons between us and the local community. So what we sort of envision is that local communities have local knowledge of Shieldtail snakes, which are very, very critical, right? And Shieldtail ambassadors help us connect with these local communities so that we are able to tap into this local knowledge and also give back to these local communities in terms of sharing our knowledge with them, right? So it's sort of like a bilateral, between local communities and researchers, right? And as part of the project, now we're doing a lot of social media outreach. We have an Instagram page called Shieldtails, where we put up a lot of posts about Shieldtails. We have a lot of engaging activities, quizzes, and so on. Right? We also have a lot of on-site outreach programs where we focus on forest department staff and now largely estate and plantation workers, students, and so on. Right. And all of this is ultimately leading to them sharing their information, their sightings with us, right? So how this works is anyone who's going out, either walking through these estates or hiking into some place, they find a Shieldtail, they take a photograph either on their cameras or mobile phones, and they upload it on citizen science platforms where we have our own portal. You can India Biodiversity Portal or iNaturalist. We also have a separate EpiCollect page where the data remains with us and is not made public. And all of this data is sort of curated by a board researcher, right? So I spend every week I spend several hours just looking at photographs of Shieldtail snakes and trying to get what data we can from these observations, right? And what data we get, of course, is one species, what kind of species it is. Many times, of course, we don't get this, right? But to a large extent, to some extent, we are able to identify species from photographs. We also have date and time, but more importantly, we get what occurrence data. This is GPS coordinates of where this species was found. Right. And all of this goes into the Shieldtail Mapping Project database. So every single observation that is uploaded here gets curated and then goes into. Right. And this is what the data looks like so far, right? Currently, we've compiled over 1,200 plus Shieldtail observations, right? Of these, just about 133 are verifiable records from actual research, right? So you can see the number of observations that are coming in, right? And just to give you a sense, so this is how the number of observations have increased over, say, the last 20 years. And Shieldtail Mapping Project started in 2021, and from 2021, you can see that there is a huge spike in the number of observations. So clearly, some of the outreach that we are doing is getting back to us in terms of data. Right. Just to give you a comparison of how significant this number is, right? I've been doing Shieldtail field work for more than 10 years. In 10 years, this is the amount of field observations that I have made. That is just 20% of this data set. That means of this 1,000 plus odd Shieldtail observations, only 200 observations I have made personally over the last 10 years, right? So you can just imagine the amount of data that we've been able to get just in, say, about 3 years, right? And this data is coming from different places. A large majority of the data is coming from Karnataka. Even though Kerala and Tamil Nadu have higher diversity of Shieldtail snakes, still comparatively lower amount of data inflow. But this is also because a lot of our our outreach is sort of concentrated within Karnataka and also Maharashtra to a large extent. Right? So we're getting data from across the distribution of Shieldtail snakes. Right. And with this data, what we're able to do is we're able to identify, right? So you see these boxes where there's no coloration, and this is where there are no Shieldtail observations. It doesn't mean that there are no Shieldtails over there, right? It's just that these Shieldtails haven't been sampled, right? So now we've been able to identify these sampling gaps and go to these regions and have region-specific, outreach programs. For example, just over a year back, there were absolutely no observations from Goa, right? But we've then gone and had several outreach programs in Goa, which has led to many Shieldtail sightings in Goa in the recent past. Right? This has also led us to find some really interesting snakes. This is an example of a Shieldtail snake called the golden Shieldtail. It was described by Beddoe in 1880, and after that, never seen. Right? Not because we haven't looked for it; we've been looking for it since. But because of the Shieldtail Mapping Project's outreach, we had some forest guards come and tell us, this might be a better location to look for. And after 142 years, we finally managed to find a population of the snake. Right? So we made some really huge, really interesting finds. We're also getting a deeper understanding of distribution. This is Europeltis journey. It was described from Bangalore. But now we've got points through citizen science from all across Karnataka, right? And this might, in fact, be one of the most widely distributed Shieldtails in the Western Ghats. Right? We're also getting information on their ecology, right? So this is an example, a few examples of arthropods feeding on Shieldtail snakes, which we didn't know until very recently, right? And basically, two of our citizen scientists shared their observations, and we were able to help them publish their observations in the journal, right? So we're here at a position to help citizens themselves become, scientists and publish their observations. We've also stumbled across the new species just because of citizen science. Earlier, I told you about this example of Herpetologist, right? Just last year, we described the species called Herpetologist aghanaashini. So before this, our understanding of the genus Herpetologist was that the distribution ended in Wayanad. We did not know that it extended all the way to Karnataka, right? And in July 2021, one of our citizen scientists, Bhargavi, she uploaded an image of this snake on our portal, right? And that's when we realized, okay, this genus is also found in Karnataka, and we have to go looking for it. Right? Later on, another naturalist, Daimler, found another population in Aghanaashini, and we were able to go sample it, and that is one of the reasons why we described this as the new species. So you see that citizens are directly contributing to our understanding of taxonomy as well. Right. Now, the main goal of the Shieldtail Mapping Project was to do conservation assessments. Right. So for that, we need data-driven distribution maps. Right. So what we did is we selected 12 species for which we had good enough data, combining our observations plus citizen science observations and so on. And with this, with these data points, we started to build more accurate range maps, right? And with these range maps, now we can go back and reassess the conservation status, right? Now, how does IUCN assess make these conservation assessments, right? So one way that it does is it uses a restricted geographic range, and it uses two important statistics or metrics, right? One is extent of occurrence, right? So take, for example, this is a range, this is some forest, and each of this is a data point, right? And these gray regions are their area, the habitat in which you find this, right? So now the extent of occurrence is basically a minimum convex polygon around all of these points. So it's the minimum area that encompasses all of these points, right? That is basically EOO. Right. They also use another metric called area of occupancy. Right. So what this does is basically we grid up the entire range into two kilometer grids. So each of this square is a 2 square kilometer area. Right. And then we count. And then we count how many of these areas we've actually seen these species, and we calculate this area, and this is what becomes the area of occupancy, right? Now IUCN has come up with thresholds, right? If your EOO is less than 100 square kilometers, critically endangered. If it is less than 5,000 square kilometers, it's endangered, and so on. Right? How they came up with this, I have no idea, but this is what they follow. Right. So what we did is, okay, now that we have better data, we built these range maps. And what we did is we overlaid them onto the range maps of IUCN and GUARD. Right? And just by looking at this, so our range map is the green, and IUCN is pink, and GUARD is the yellow. Right? And for each of the species, you see that there is a huge difference between ours and expert drawn sort of range maps. Right? And when you actually compare this IUCN EOOs, right, what we see is it's hugely off, right? In many cases, IUCN has underestimated the geography. That means the range is smaller than it actually is, and in most cases, it has overestimated the geographic range. In the sense, the actual range is smaller, but IUCN has a much wider range. Now, given that IUCN uses this for conservation assessment, you can just imagine how errors like this will affect where a species goes into, whether it goes into endangered or critically endangered. So, right? Now, not just are we able to build these range maps, we are also now using GIS information, right? We're taking satellite information and trying to sort of extract what the threats are within these range maps, right? So let me give you an example. So this is a species called Melanophilium carey. Currently, IUCN classifies it as endangered, right? So one of the threat parameters which we're taking is the overlap with protected areas. The protected wildlife sanctuaries and national parks, right? And these areas inherently have sort of some kind of legal protection. So what we're doing is we're placing the range map over it, and these red ones are the protected areas, right? And we are calculating, okay, how much of this area is within a protected area and how much of it is outside. We're also calculating the tree cover loss, right? So again, within this area, how much tree cover has been lost in the last 20 years, right? So we've calculated that again based on satellite data. We're also calculating something called as human footprint index. Now, human footprint index is basically a chutney of multiple different threats, right? Say it includes road densities, it includes built-up area, how many buildings are there, it includes population density, it includes nightlight, and so on and so forth. So it's a chutney of 8 such parameters, right? And so we calculate what is the average human footprint within this geographic range as well, right? And based on this, we can actually put all of these into a graph. Now, I don't want you to look at these graphs in super detail or anything, but I'm just going to give you two examples. So this is a species called Europedis rurolineata, which is a fairly small sized, it has a very small geographic range, somewhere around 300 square kilometers. But if you look at this, almost 95% of its geographic range is within a protected area, right? Also, it has comparatively less human footprint and very less tree cover loss, right? So the species like this, even though it has a very small geographic range, is not really of conservation importance here because, I mean, there are not a lot of threats. But if you look at a species like this, Rhinophis melanoleucus, you see that it has a very small geographic range. But apart from that, 0% of it is within protected areas, right? So this entire species is found outside protected area. It has one of the highest human footprints, although tree cover loss is fairly low, right? So if you compare this, you can intuitively just say that a species like this would need more conservation protection or conservation importance than a species like this, right? So that's sort of what we're trying to do here, right? In addition to this, we can also look at road mortality, right? Now I'm going to show you some very distressing images, so please bear with me here. Now many species end up dying up on the roads, especially during the monsoon. They come out onto the roads and they get run over by vehicles, right? Now our data set also has a lot of data on roadkill, so we can actually sort of look at which species seem to be more sort of vulnerable to roadkill. This can also be added in as a threat level, right? And now we can include all of these threats, right? The geographic range and all of these threats to sort of come up with this a cumulative threat score, right? So that's basically this value over here, right? I'm not going to discuss how we got this number. That's because we're still sort of developing this new index, right? And trying to see what we can do here. But yeah, so basically what we're trying to do is combine all of these threats and see which has a higher threat. So you can see that a species like Rhinophis melanoleucus has a super high threat, but on the other hand, a species like Melanophilium kerei has a fairly low threat. Right. And so this is the current IUCN state that these species are classified into, right? But you notice that for some species, you can actually downlist it, right? Something that is orange here is becoming green, right? But on the other hand, what you notice is that most of the species that are either green or gray are becoming more hotter colors. They're becoming yellow, which is vulnerable, endangered, or critically endangered, right? Which is basically telling us that when we add more data, right, we can make more informed conservation assessments, right, and know where to prioritize our conservation. Right. So the take-home message from all of this is that the underground environment holds remarkable diversity, right? But because of all the specializations that they have, they're extremely vulnerable to extinctions. But then there is a general lack of basic data, right? And we really need a collective approach towards generating data and towards conservation itself, especially when it comes to such, rare or secretive, right? So with the effort of large group of people, now we are sort of able to make some changes here and there, or suggest some changes here and there, and and we're going to be counting more on such citizen science driven approaches. Of course, I have to thank a lot of people for being part of this work. Right. So currently, the Shieldtail Mapping Project is run by Anuj, and then we have Sanskriti, who's our social media manager. We have Praveen Hechen, who works for Amazon and was instrumental in setting all these up. We have 150 plus contributors. And of course, we have to thank our funding agencies, many organizations, and finally, I have to thank the Aghanaashini Foundation. I've been told that it's been exactly 1 year since they started their webinar series, and the first talk that they had was again on Shieldtail did, And exactly after 1 year, we are ending with another Shieldtail talk. And I'm glad that we're now able to show some data that we've generated over this 1 year. Right. Anyway, thank you, and I'd be happy to take questions if there are any.
Thank you for your wonderful session. Now the session is open for discussion. If participants have any doubt, you can ask by unmuting yourself or by putting messages in the chat box.